is Sara Howden. I'm from the Murdoch Children's Research Institute in Melbourne, Australia. I'd like to start by thanking the organisers for the opportunity to talk about our work today. So the vast majority of our research is centred around human pluripotent stem cells, or more specifically induced pluripotent stem cells, which were first described over 15 years ago now by two independent groups led by and James Thompson. And in their groundbreaking work, they were able to show that terminally differentiated cells could be reprogrammed back to a pluripotent state simply by forcing the expression of a defined set of transcription factors. And essentially, RBSCs are equivalent to the embryonic stem cells in that they can be cultured indefinitely in a pluripotent state, or given the right cues, they can also give rise to all the different cell types that make up the human body. And the enormous potential of these cells lies in the fact that they can be derived essentially from any individual, and as such, they have huge potential for a variety of downstream applications, including disease modelling, drug discovery and toxicology platforms, and as well as holding enormous potential for developing cellular therapies for a wide range of currently intractable disorders. So our group, led by Professor Melissa Little, has pioneered the development of protocols for the generation of kidney tissue from human pluripotent stem cells. And these protocols are largely based on our understanding of how the kidney actually develops in vivo, and it involves guiding the stem cells in a stepwise fashion to primitive streak, and then onto intermediate mesoderm, at which point the cells are then aggregated and cultured in an air-liquid interface. And at the end of this process, these three-dimensional structures we call kidney organoids contain many of the compartments or cell types that we expect to see in a developing kidney, including fully segmented nephrons with distal and proximal tubules, podocytes, and as well as a renal stroma and developing vasculature. So while there are some key differences, kidney organoids represent a pretty reasonable model of the developing kidney in vivo. And this means that it can be used in a spectrum of exciting downstream applications, ranging from furthering our understanding of human development to tox and drug screening platforms, and hopefully in the not too distant future, even the development of cellular therapies for patients living with kidney disease. But a big focus of our research to date has been centred around the use of kidney organoids to model disease. So the use of organoids for modelling kidney disease typically involves a workflow that starts with a patient or a group of patients for whom a genetic variant of interest has been identified. And this may or may not be previously known to be associated with disease. iPS cells are then derived from the patient, most often a blood sample, and then the genetic variant is typically corrected using commonly used gene editing tools such as CRISPR-Cas9. And this is important because it enables the generation of matched isogenic controls, which are genetically identical to the patient other than the genetic variant of interest. And so this enables us to attribute any functional variation between a patient and a gene-corrected sample purely to the variant of interest, rather than differences in genetic background. So the organoids can then be, the iPSCs can then be differentiated into organoids, and then we can interrogate these using various assays in the lab to try and gain a better understanding of the molecular mechanisms that underpin specific types of kidney disease. And today, our group has published a number of studies where we have successfully used organoids to model diseases such as nephropathitis and nephrotic syndrome. However, kidney organoids in their current form are not actually ideal for modelling autosomal recessive polycystic kidney disease, or ARPKD, as they typically don't contain a ureteric epithelium or collecting duct. And obviously this is the main site of pathogenesis in ARPKD patients. And we did actually believe that organoids did contain a ureteric epithelium, due to the presence of an extensive network of epithelial cells that express GATA3. And this is a known marker of ureteric epithelium. And you can see this quite clearly in this image here, which is of a live organoid that had been generated from iPS cells that harbor a blue fluorescent protein tagged to a gene that's highly expressed in podocytes, as well as a red fluorescent protein tagged to GATA3. So to interrogate the identity of these cells further, we actually isolated the GATA3 positive epithelium by fluorescent activated cell sorting, and then we analyzed their transcriptional profile by RNA-seq. And we then asked the question, what do these cells most closely correlate with when directly compared to developing kidney, human kidney tissue in vivo? And we're actually in a unique position to do this thanks to our collaborator, Jason Spence, who shared his extensive single-cell RNA-seq data set with us, which had been generated from human fetal kidneys at 14 to 6 weeks gestation. And as illustrated in this heat map here, this analysis revealed that the GATA3 expressing cells in kidney organoids are actually far more closely aligned with a distal nephron identity and not ureteric epithelium. However, when we cultured these GATA3 positive cells in conditions that had previously been shown to support ureteric epithelium isolated from fetal mouse, something very interesting occurred. Over the course of 1 to 2 weeks, we observed that these cells could actually form structures with a very similar morphology to those formed from those mouse ureteric bud cells that had been isolated from fetal mice, as shown in these images that had been taken from that study by the Yanagawa group. And we also performed some immunohistochemistry analyses on these structures and found that they express commonly known markers that identify ureteric epithelium, such as cytokeratin 8 and calbinin and SOX9. And because these structures, which we went on to call as induced ureteric epithelium or induced or IUE cultures, so they were initially established from the GATA3 epithelium that we had FACS purified from organoids. We really wanted to know if we could establish these cultures in the absence of this step. So in these experiments, we took kidney organoids, again from our fluorescent reporter iPS cells, and we dissociated them and then plated them at low density in these same UE promoting conditions described earlier. And after 1 to 2 weeks, we observed the appearance of these budding structures that showed a very similar morphology to those that we observed in the sorted cultures. And we also showed that these IUE structures could reform after dissociation to single cell and reseeding, and even they were even able to form after cryopreservation and thawing. And when we compare the transcriptome profile of IUE cultures directly to our human fetal kidney data set, we see that these cells have clearly shifted away from a distal nephron identity and towards a strong ureteric tip identity. And importantly, we could also show that this identity is maintained even after passaging, cryopreservation, and thawing. So single-cell transcriptome analysis of these cultures also confirmed that the majority of cells do in fact identify as ureteric tip, although this analysis also revealed that a small cluster of cells seem to hold on to that initial distal nephron identity, and there are additional clusters that also seem to have a more differentiated collecting duct identity. And we can illustrate this point further using feature plots for some key genes, such as RET and Wnt11. These are shown here in green, which are known tip markers. And you can see these are broadly expressed in our your IUE cultures, whereas the more definitive stalk markers shown here in green were expressed in far fewer cells. So given the majority of cells in these cultures have a ureteric tip identity, and these are the progenitor cells of the more mature collecting duct, we were next curious as to whether we could differentiate these cells further by removing growth factors that promote tip proliferation, such as GDNF and Wnt, and then adding factors such as vasopressin and aldosterone, which are known to support collecting duct differentiation. And when we profile these cultures by RNA-seq, and particularly those cultured in the presence of vasopressin and aldosterone, that is exactly what we observed, a clear shift away from ureteric tip towards a more differentiated stalk or collecting duct identity. So we, of course, are very interested to know if these cultures could now be used to model diseases that affect the collecting duct, such as ARPKD. So to do this, we initially started with our fluorescent reporter iPS cells and used CRISPR-Cas9 to generate frame-shifting knockout mutations, the very 5' end of the PKHD1 gene. Of course, this is the gene that is most commonly mutated in patients with ARPKD. And then we established our IUE and stalk cultures from both wild type and PKHD1 null iPS cells, and then monitored them by fluorescent microscopy. And we were delighted to see that the stalk cultures established from the mutant iPS cells spontaneously formed these large dilated structures that reminiscent of the cysts that we typically see in the collecting ducts of ARPKD patients. And we also performed a quantitative analysis on these structures, which revealed a significant increase in both cyst area and perimeter in the PKHD1 null cultures compared to their wild type counterparts. And while these dilated structures form spontaneously in PKHD1 null cultures, we also showed that these structures rapidly responded when challenged with forskolin, which is illustrated in these time-lapse videos, which were taken over just a few hours. So to investigate whether we can apply this workflow to patient samples, we have obtained blood draws from 3 children now who were diagnosed with ARPKD at the Royal Children's Hospital, where we are based. And all 3 patients have known mutations in PKHD1, which we identified by genome sequencing. And some of these mutations have previously been associated with ARPKD, such as this serine to methionine change in amino acid 36, which is quite common in patients with ARPKD. And we've now generated sets of iPSCs and isogenic controls where we've used CRISPR-Cas9 gene editing to correct patient mutations, which we have highlighted in green in this table here. And these sets of patient and gene-corrected stem cells are now being differentiated in vitro and assessed for the formation of the dilated cyst-like structures that we observed previously in the PKHD1 null stalk cultures. This is just some preliminary findings of one such experiment using patient samples. I believe this is patient 3 in the table that I just showed. And in this example, we observed cyst dilation in stalk cultures derived from patient iPSCs relative to cultures derived from the gene-corrected controls. And this was especially evident after challenging the cells with forskolin. So these preliminary findings may suggest that perhaps in cells with less severe mutations in PKHD1, such as the missense mutations in this particular patient, that environmental stress in combination with the genetic aberration may be required to model ARPKD in vitro. So we're now establishing IUE cultures from the additional sets of patients and isogenic controls, and we're actually in the process of collecting samples for phosphoproteomic analyses which we believe will be extremely beneficial for furthering our understanding of the key molecular pathways that underlie cyst formation in ARPKD. And we believe that these analyses will also be critical to help us to design a targeted screen to identify compounds that may prevent or even reverse cyst formation, which may be of particular benefit to patients living with ARPKD. So I'll just like to finish by acknowledging the people who were involved in this work. Of course, Melissa Little, my mentor, and the people listed beneath have all been involved in this particular project. And our collaborators in the Spence Lab, and of course our funding bodies, particularly the PKD Foundation in the USA and PKD Foundation in Australia. This work is also supported by the NHMRC in Australia. Thank you for your attention.
Okay, thank you. I don't think Sara is available for live Q&A, so we will move on to the next presentation. And the presentation is from Dr. Feng Qian from the University of Maryland. And I don't know the title of his talk, but he'll tell us. Oh, there it is.
Okay. I thank the organizer and PKD Foundation for inviting me to present our work. Okay, as we have heard from last talk, the ARPKD is really a very severe form affecting mostly fetus or neonates. And the disease is really, the key kidney pathology is really involved dilation of collecting duct. So in order to understand the mechanism of this cyst formation in ARPKD, and various autologous mouse model of ARPKD has been generated. And these are the mouse containing mutation in the causative gene Ph.D gene. Okay, so in contrast to severe human form, phenotype found in human ARPKD or in the ADPKD mouse model, all these mouse model of ARPKD is really unexpectedly mild in the kidney. So we found no or very limited cystic dilation in early stages. And instead they found a very mild cystic dilation in the proximal tubule or in collecting duct in high age. And instead, these mouse model really develop only the biliary cysts or in fibrosis in the liver. Occasionally, they find also a cystic dilation in the pancreas in the old age. But bottom line is that these mouse does not really tell us too much about the pathogenesis because they live a normal life and they don't have cyst formation. Okay. Yeah. And the PKD1 gene encode a protein called fibrocystin or polydactin. And this is a very large single transmembrane domain protein. And the study using recombinant protein or using epitope-tagged PKD1 mouse model showed that this protein is really undergo a very complex processing. And the protein is cleaved at extracellular domain and resulting into a fragment is very similar to like polycystin 1. Yeah, and they can remain together, these two pieces. And the proteins also get cleaved at or near the the transmembrane domain, resulting in the formation of the various intracellular fragment called ICDs. And interestingly, that the C-terminal tail of the protein contain various motif. Particularly interesting is the CTS. This stands for ciliary targeting sequence, which actually make the protein to go to the cilium. Yeah. And the protein also in this area also contain a NLS, which is actually stands for nuclear localization sequence, make really the potential form of ICD go to the nucleus. But what really unknown at that time point is what happened to the native endogenous fibrocystin protein. How are they processed? And if they do, and where do these protein go? And what kind of function they may have. So we start our study in ARPKD really by asking those questions. So we want to know what happened to the endogenous native protein. In order to do that, we developed a set of antibodies against mouse fibrocystin protein. That include a protein directed to different places in the protein domain. So we're using this panel of antibody to characterize the pattern in the developing kidney. In this case, is this is actually a Western blot using kidney, wild-type kidney. Wild-type kidney as a control is the knockout kidney. And we also use a HA-tagged form of a mouse model provided by, actually generated by Terry Watnick. And as you see here, that this antibody, E1 antibody, really generate, give you a set of a band, really a lot of bands absent in the knockout mouse. And the sum of the band is known. For example, this one is full length. This one is this is the PTM. Yeah. But interestingly, we found also a much smaller band right here in this area. Okay, and this band occur also in this HA tag form, with the exception that this band is run a little bit slower. And this is expected because you have the HA tag on it. And these 3 bands is recognized only by the what they call this HA antibody. So to map where this cleavage might happen, we use a ladder of fragment of polydactin or fibrocystin protein, which start at a different position from the C-terminal tail. Using this ladder, we were able to really come up with this model that this endogenous protein of polydactin really undergoes a complex processing, resulting in at least 3 transmembrane domain protein, as well as, I have to go, sorry about this, yeah, as well as 3 soluble ICDs. And of note that we did actually in silico study, and we found that these polydactin protein contain an MTS, which stands for mitochondrial targeting sequence. And of note, interesting is that this one of the ICDs, which is here, ICD15, contain a start with MTS. Yeah, and interesting is that this part of a sequence contained within ICD15 is encoded by the last exon, exon 67. And this really, this is just give you in a sequence to tell you that the ICD15, let's see, yeah, 15 and 15, contain its very N-terminus MTS, followed by NTS, NLS, and this whole sequence is really encoded by the last exon. Okay, so having said that, so the question is, okay, you have a new form of polydactin protein, ICD15, and containing MTS. The question is this really functionally important for this protein to be translocated into mitochondria? In order to test this hypothesis, we actually generated ICD15 construct fused to GFP protein. Okay, and so here these are the 2 constructs. Yeah, can you see this? This is actually the human. This is the mouse version of it. As a control, we actually make a mutant form of mouse ICD15 containing a point mutation with predicted reduced MTS score. Okay, so this is actually when you transfect this construct into IMCD cell, and you found that for human and for mouse construct, if you look at the transfected cell, and they actually give you a pattern typical for a mitochondria. So in other words, this construct, if wild-type construct, whether it's human or mouse, they are translocated into mitochondria. And in contrast, if you look at the mutant form, they do not. Instead, they actually go to nucleus, as you would expect, because they do have the NLS sequence on it. So maybe they go to both places, and depending on, they could go to both places, but if you knock out one location, you may enhance the other one. So we basically, to further confirm that this is really true, we did another assay called the split GIP assay. Yeah. So I'm not going to go to detail how that works, but basically the only thing which you need to know here is that every time when this the ICD actually go to mitochondria, you're going to see a yellow mitochondria. And for this assay, we actually use, again, we use either a mouse, human wild type, ICD15, and as a control, we use 2 different mutant form of human ICD15. They should actually have a predicted reduction in mitochondrial translocation. Okay, so this assay is, as you can see here, both human and mouse, the mouse and human, they actually translocated into mitochondria, as you can see by yellow mitochondria. In contrast, the mutant form of it, yeah, they do not. Instead, they form a fuzzy pattern. And this is just another control. So far I have shown you that polydactyl native form, they produce multiple bands, small band, and from C-terminal tail, and they go to mitochondria. All right. So now the next question is, what do they do? So, okay, what do they do? So in order to answer that question, we actually collaborated with Alessandro Boletta, and we knocked out PKHD1 in IMCD cell and did the seahorse study. And we found that if you knock out PKHD1, they reduce, you reduce really the basal respiration and maximal respiration. Okay, now we ask the question, what happens if you overexpress ICD15? Does it actually change the mitochondrial function? So what we do is we transfect either wild type or mutant form into either MCD wild type or the knockout mice cell, yeah, and do the seahorse assay. So what we actually found is that, okay, here, this is a better one. Yeah, so what we found is if you overexpress the wild type ICD into the wild type MCD cell, it actually enhances mitochondrial function. But if you look at the wild type form, the mutant form, they do not, suggesting the enhancement of this ICD15's function in mitochondria depends on the mitochondrial localization. You need to get it into the mitochondria in order for them to function. And similar finding was found if you do the same thing in the PKG knockout mice. Okay, so next question is, is it important at all? Are they actually involved in cystogenesis? Yeah, because we know that the knockout mice, they don't, yeah. So in order to show that, we actually did the epigenetic study, yeah. So that means you can breed this, you can breed a, sorry, yeah. Yeah, I'm gonna skip this now because of the time, yeah. So basically they form a abnormal mitochondrial morphology in vivo, despite of the fact that they do not have cysts. So that's indicate that these, although they looks normal, but they're not, yeah. So because they have mitochondrial defect in the PKHD1 knockout mice. Okay, now is the question is, is it important for cytogenesis? We do that in the epigenetic study. So we actually breed the knockout mice with the PKD1 VV mouse, as we have heard. And in both situations, at the P0 time, they're not cystic. But if you bring them together, what you find is actually the combined mutation really form a very severe cystogenesis in the kidney. You can see that in P0, you can see that even before birth, suggesting that PKHD1 really provide a defect, procystic condition on top of a PKD1, and therefore causing severe cystogenesis. This looks basically like a PKD1 and KO phenotype in the kidney. And final data, what I'm going to show you is the question is, okay, so PKHD1 does have some role to play, important role in cystogenesis through we think it's through mitochondria. But in order to show that, we actually need to ask the question, what happened to this C-terminal tail, which go to mitochondria. Here we go, yeah. So in order to answer this question, we were very fortunate because Terry Watnick actually provided us this particular mouse, a mouse deleting specifically the last exon 67. So we had to use this mouse model and do the genetic study in combination with the PKD1-VV. We show that the protein is made, yeah, so this mouse is useful for us. And this is basically show you that that the combination of these delta 67 mouse together with the VV enhance cystogenesis in the kidney as well. Although it's not as severe as the knockout, but they do. Yeah. So I'm going to actually summarize. So what I'm really telling you is that proteolytic cleavage of polydactin or fibrocystin generate a ICD-15 in vivo. And so this fragment enhance mitochondrial function in oxygen consumption, respiration, through mitochondrial translocation via unmasked mitochondrial targeting sequence. And PKHD1 knockout kidney tubule display significant ultrastructural alteration in mitochondria, even in the absence of cystic form. And PKHD1 translocation induced PKD development in a non-cleavable PKD1 VV mouse model resembling PKD1 knockout mice. And lastly, I showed you the deletion of that exon, which go to mitochondria, can also enhance cystogenesis in the PKD1 VV mouse kidney. I'm going to make some conclusion. I thought maybe important. Number one is PKHD1 plays a critical role in regulating proper structure and function of mitochondria in renal and pancreatic epithelial cell, which I didn't show you, but they also have the similar pattern. And the regulation of mitochondrial function by FPC involved proteolytic cleavage and release of ICD-15, followed by its translocation into mitochondria. And in this polydactyl inactivation generate a pro-cystic condition, not cystic, but pro-cystic condition that interact with or converge on the PKD-dependent cystic pathway during kidney development. And finally, fibrocystin suppress cystic dilation in part through its mitochondrial ICD-15 when PKD1 function is compromised. I'm going to stop here and thank all the collaborators and also for the PKD Foundation for funding. I'm going to stop here and take any questions if you have.
Thank you, Feng. Beautiful presentation. Questions? Maybe I can start. Have you done the ICD-15 O expression in a PKD1 null cell line? And does that rescue any of the defects?
Yeah, that could be. No, I haven't done that, but that could be one of the future directions. Yeah.
Okay.
Yeah.
Very interesting presentation, wonderful data. Can you comment on what is happening in the mitochondria? How could this, protein cause contribute to the increased respiration?
Yeah, thank you for the good question. We have actually, Monica, yeah, she has a very good, very nice poster yesterday or today, and we have a nice discussion. And based on your data, I would suggest it could be that the CTT, that called ICD-15, may actually go to the mitochondria. Either they interact with CTT of polycystin 1, we need to test that. But the question is, how do they interact with the mitochondria? One possibility would be they may interact with the component of the mitochondria, either using your uncoupler. Yeah, just one example. Or using a VDAC, So basically, they may actually interact and affect the mitochondrial function through some component. I think the proteomics study will actually maybe give us some information for the future. Thank you.
Thank you for the talk. That was exciting. I have two quick questions. One is, do you know why it prefers to translocate to mitochondria over the nucleus? And the second question would be, if it's increasing mitochondrial flux or respiratory flux, does it also mean there is an increase in oxidative stress?
Yeah,
Thank you.
That is the hypothesis. Could be. We have not tested yet, but it could be done, yeah. In terms of the why MTS versus nuclear translocation, right? This is actually interesting because if you look at the sequence, these two sequences overlap. So they could be somehow mutually regulated. Now, I don't know what, maybe by phosphorylation, because there's some consensus site right there, yeah. So it could be by modification, could be affect the pathway, favor the translocation either to mitochondria or to nucleus. So it could be co-regulated and could be even crosstalk between the two organelles. But further work needs to be done.
And I'll finish up with the last question. Are you making ICD-15 transgenic mouse just like the CTT, PC1 CTT, like from Mike Kaplan's group?
Yeah, I'm thinking about to submit a PKD foundation.
All right, thank you so much,
Thank
Frank.
you.
All right, our next presenter is Christopher Banek from the University of Arizona, and his title is Role of Renal Nerves in a Preclinical Model of Advanced ARPKD.
Okay. All right. I was told to provide a disclaimer, so this is only for educational purposes, obviously, and I have no financial disclosures at this time, unfortunately. I was also told to give you a background of my training. I'm going to skip over that. I don't want to bore you to death. So we'll just dive right in. And so this audience obviously needs no introduction into ARPKD, PKD, ADPKD, nor the severe limitations in the treatments available for these patients. But the primary treatments available are renal transplantation, but those are often few and far between, not frequently available, so they are oftentimes devoted only to hemodialysis while they wait. In 2018, tolvaptan emerged as an FDA-approved treatment for the mitigation of cystogenesis in ADPKD, and current clinical trials are in place or ongoing for ARPKD. While promising in the blunting of cystogenesis, there are negative consequences to chronic V2 blockade, and that is polyuria and polydipsia. So your lifestyle is dramatically changed. You are constantly your life constantly revolves around the bathroom, and your sleep is disrupted, obviously, too. So obviously, further research in the development of new modalities and maybe new targets for PKD treatment is needed. So I had a very nice lead-in with other talks, so I just want to highlight a few of the phenotypes that I think are very crucial in where I'm going with this and the rest of the talk. And that is hypertension, renal inflammation, and fibrosis, and lower flank pain are commonly associated with ADPKD as well as ARPKD. Arginine vasopressin is also elevated in many of these cases. It's not really clear why, though. So vasopressin, aside from regulated by plasma osmolality, is also regulated by sensory nerves from the kidney. The nerves at the kidney relaying information from kidney back to brain, i.e. interoception. When stimulated, you can cause an increase in vasopressin production and release. And so I assume renal neuroanatomy is not well known amongst this crowd, and so I'll just briefly cover it. So the nerves innervating the kidney are sympathetic and spinal afferents. There's very limited evidence for parasympathetic innervation of the kidney. It's largely sympathetic. I want to stress that. So we have the sympathetic side here in the purple. They innervate all throughout the cortex, the medulla, and the pelvis. They're distributed all throughout the kidney. And the spinal afferents that are innervating kidney are also distributed all throughout the cortex, the medulla, and the pelvis. And note that they all are traveling along the renal artery, making it very easy to access in our surgeries. The neurophysiology is pretty well defined for the sympathetic contribution for sympathetic regulation of the renin-angiotensin system through renin release, sodium retention at the proximal tubule, as well as regulation of renal vascular resistance, whereas the afferent neurophysiology is not really well sussed out right now. That's a very active area in the field. There's a lot of data to suggest that they are sympathoexcitatory when stimulated. They reflex back to the brain and cause a peripheral increase in total sympathetic tone. But there's also evidence for a contralateral suppression of renal sympathetic nerve activity that's a spinal reflex, known as the renal-renal reflex that was pioneered by Ulla Kopp. Nevertheless, I want to dive into how I got into PKD, because I come from the cardiovascular field, the neurophysiology field. And my previous work was in docosalt hypertension. This is a model that's severely hypertensive as well as mediated by vasopressin activity. Okay? So we target the renal nerves through a technique known as renal denervation. We go in, cut nerves, okay? And if we do that, renal denervation prior to the initiation of docosal hypertension, compared to the sham control here that you see is becoming hypertensive over the 3 weeks, the red group here, TRDNX, has a nice attenuated response. There's an attenuated hypertensive response. Notably, if we target just the renal sensory nerves, but leave sympathetics intact, we get an identical mitigating effect in the development of hypertension. And that in the more clinically relevant paradigm is patient presents with hypertension. If we intervene after they're already hypertensive with either total denervation or afferent denervation, we lower pressure identically. And this suggests that afferents are very crucial in the development and maintenance of hypertension. In addition to the hypertension, we've also seen an attenuating effect of renal inflammation, these pro-inflammatory cytokines that are upregulated with docosal hypertension are abated with total denervation and to a lesser extent afferent denervation. And notably, we were the first to show that there is an elevated sensory nerve activity in these hypertensive animals, renal sensory nerve activity, compared to the normotensive vehicle controls. Sorry. And so a lot of these overlying symptoms that I just raised, the hypertension, the renal inflammation, and lower flank pain, I've associated also with increased afferent nerve activity. I simply had the hypothesis that in PKD, we have very similar phenotype. So we needed to quantify whether or not PKD presents also with elevated sensory nerve activity, as well as do renal nerves contribute to the progression of cystogenesis in PKD. And so I largely work with rats because I hate working with mice, and the only model that was available in rats was the PCK model, which is a model of ARPKD. It's a loss of function point mutation in PCK, which is orthologous to the PKHD1 gene, which we just heard about in the previous talk. So this making a good model for ARPKD. Cysts develop in utero and advance very quickly. The data I'm going to show you today was performed by my Ph.D. student, Madeleine Gaudier. And so we went in and recorded afferent nerve activity, first of all, just to demonstrate whether or not afferent nerve activity is indeed elevated compared to non-cystic controls. And how we do that is we anesthetize an animal, hook a renal nerve bundle, cut the central end of that, and record only afferent nerve tone. After the baseline recording of that, we stimulate those afferent nerves with a TRPV1 activator or TRPV1 agonist known as capsaicin, that spicy little ingredient in chili peppers. We get a maximal activation of those afferents, and now we're going to express that afferent nerve activity as a percentage of max. And so indeed, as we hypothesized, the cystic animals at 10 weeks of age show an elevated afferent nerve activity at rest compared to our non-cystic controls, and it's shared relatively similarly between males and females. One of the coolest parts about this is that we took those cystic kidneys and we correlated their cystic index to their afferent nerve activity, and we saw a direct correlation to increased cystic index or advancement of cystogenesis correlates to an increase in afferent nerve activity. But we still have this kind of chicken or egg question right now, and this is an active area in our lab. But let's dive into the intervention, renal denervation, because that's what we do. We go in, cut nerves. And so we're going to look at it in two paradigms. We're going to be looking at the early and late phase intervention. So let's start off with the first phase here. And so right after weaning, we go in and perform the renal denervations. At week 4, follow them out 6 weeks, and at week 10, we collect tissues for further analysis. We implant telemeters at age 8 to monitor blood pressure responses. And very briefly, we go in and actually dissect off the renal nerves to perform a total renal denervation. This gets rid of sympathetic and sensory nerves. But afferent denervation is done chemically, where we wrap the renal artery with a very, very concentrated solution of capsaicin. And this targets those nerves that express TRPV1, causing a huge calcium influx into those cells, poisons them, kills them, but leaves sympathetic nerves intact. So 6 weeks post-denervation, looking at blood pressure, we saw, not very surprisingly, total denervation, lowered blood pressure. Very cool. We did not see that effect with afferent denervation. But arguably, the coolest part of the study is that we saw a mitigating effect of the cystogenesis from week 4 to week 10 was attenuated with total denervation and identically with afferent denervation. So this really alerted us to the idea that afferent renal nerves contribute, at least in part, to the cystogenesis in this model in the early phase. And I just want to compare this to a previous study using a V2 blockade over the same timeline. Early intervention followed out to week 10. They also saw a similar attenuating effect of cystogenesis with chronic V2 blockade, but that's also associated with increased water intake, increased urine output, which we did not see following renal denervation, yet we got similar results. So that was very, very promising. And so we wanted to go forward into the late phase intervention. And this is our most recent data. So we did the exact same paradigm. We intervened at week 24 where they have prominent renal cystogenesis and a decline in GFR as compared to normal values, which is around 1. So let's get to the biggest find, is that we actually saw a reduction in cyst size even at 24-week intervention out to 30 weeks. We saw total denervation and afferent denervation having a similar mitigating effect of the cystogenesis. And GFR declined over those 6 weeks in our sham treatment group, yet we did not detect a time-dependent change in GFR in either denervation group. And pointing to a possible mechanism is that serum copeptin is lower in our total denervated group. Serum copeptin is a chronic marker and estimate of vasopressin activity, and so we could potentially be disrupting vasopressin activity with total denervation and potentially, I should say, with afferent denervation, as that was not significant. But I'm trying to wrap my mind around this because this is a fairly advanced stage of the disease. We have declined kidney function and severe cystogenesis at this time, yet we have a reduction in the cyst size. We could easily just be attenuating the development over time, similar to the early intervention studies where it just continues to progress, whereas that is blunted. But we cannot rule out the idea that it remains relatively stable over those 6 weeks, and we are actually causing a decrease compared to baseline. Now, we have the data collected on ultrasound imaging over time, so we'll have that to see whether or not it changed from baseline or it was simply a mitigating effect of the progression, but this would certainly be exciting. And so this is a working hypothesis, is that the early phase of the cystogenesis and the inflammation is exciting these afferent nerves, reflexing back through an interoceptive mechanism and causing an overactivation of that vasopressin-mediated mechanism and causing a decline in kidney function. And so the conclusions I want, or the main takeaways I want to end with, is that renal denervation seems to mitigate the early and late phase cystogenesis in this model. And it may be mediated through an increase in afferent nerve activity and an increase in vasopressin activity. And so these are there are 3 active areas in our area of research right now. And so obviously this research is performed by an incredible team of mine at the University of Arizona. And so I want to thank them for all of their hard work, as well as the very, very important funding that has been provided over the last several years. Firstly, the PKD RRC with Dr. Watnick, thank you very much for that, as well as the most recent one is the PKD Foundation's Research Award. And so with that, I'll end there and take any questions at this time.
Thank you very much, Chris. Really beautiful data. Questions? Okay, Terry.
Really nice talk. I'm not a neuroscientist, so maybe this is a simple question, but do you think that it's catecholamines and that is what's working? And if so, could you like try a beta blocker or something to see if that does the same thing? Or
So are you saying that catecholamines reflexing down into the kidney? Yes. You could potentially give a beta blocker. I would really like to do that chronically in these studies. I showed you two interventions here, the total denervation and afferent denervation. Those are two relatively easy procedures to do, but the one I really want to do, it's a little bit more surgically intensive, but you can ablate the sympathetic nerves and leave afferents intact, but that's going to require some cannulation and isolation of the renal vasculature for about 30 minutes, because you need to provide 6-hydroxydopamine for about 30 minutes to poison those cells, because you need to attack that at the terminal, because that needs to be uptake at the end, rather than it's not expressed or taken up at the axon. But that's definitely on our plate right now.
Thanks so much. Great talk. Similar question: can renal angiotensin-aldosterone system have an effect on this? You are doing renal denervation, which may maybe decrease the renin, then it may contribute or the improvement.
Potentially, but I always go back to the idea of why are we getting similar effects with afferent nerves? Afferent nerves aren't contributing to renin-angiotensin regulation, at least
As
directly.
far as I remember, in renal denervation studies, when over they look, the renin levels are going down, right?
They're very mixed results
Mixed
in
results.
those,
Okay, maybe.
but they're either not changing or going down. But again, that's with total denervation, which it's very well understood that the sympathetic nerves, efferent sympathetic activity, is contributing to that, which we've almost removed from the mechanism now, since we are achieving similar results with just afferent denervation. Now, we can't rule out potential effects on sympathetic activity to the kidney after renal denervation. That certainly cannot be ruled out.
Thank you.
Yeah.
Nice.
Thank you so much for the presentation. I may missed, so how much of the blood pressure affected by your intervention, and how do you exclude, the effect is actually from low blood pressure?
Yeah, we certainly can't exclude the effect of lowering blood pressure on the effect that we saw in cystogenesis. Yeah, that would take some chronically instrumented animals to clamp pressure, yet have our denervation intact to address that question. But we can't rule that out. Yeah, but we didn't achieve a blood pressure drop with afferent denervation, which we did with the total denervation. So I guess indirectly we're kind of addressing that, but I don't feel confident in ruling that out, obviously.
Hi. If you could give capsaicin in the kidneys of these mice while they're still alive over some period of time, would you predict that it would have an effect on the amount of cyst formation?
If I could give capsaicin over time, I would not. I don't think our IACUC would allow that. But to the kidney, that would certainly cause them an incredible amount of pain. But I'm going to change that question a little bit. I'm going to say I could give chronically capsaicin, which is a TRPV1 blocker, chronically. I would hypothesize we'd get similar results. So if you want to do a pharmacological therapy like that, that would certainly be a very good experiment to address, and I think that would mitigate cyst development.
Okay, I will finish up with the last question. You just happen to work on the ARPKD model because of the convenience of the species, right?
Yeah.
But do you think the same thing would be applicable if you did this in the ADPKD models?
I'm glad you asked that question, because we are doing that right now with in collaboration with Dr. Watnick's group and Patricia Oteda, as well as Peter Harris and the ARPKD mice. And so I hope to be able to share those results very soon with you.
Great. Thank you so much, Chris. All right. So we're done for this session. And there's a research poster presentation from 11 to 12 outside. And then we'll meet back here again at 1:10 for dietary intervention and metabolism session.